Bhatnagar, embryology in relation to taxonomy. Now, in this present study, we are going to discuss the application aspects of embryology, experimental embryology, application aspects of embryology. What are those aspects? Embryology is any importance is there. Our embryology is correlating with any other branches that we are going to discuss today. Okay, this application topic is very important. We all studied the basic structure, basic embryology, and all the parts of sexual organs of the flower, development, anther, ovule, embryo, endosperm, nucellus, embryo, polyembryony, embryo development, apomixis, parthenocarpy. Now, how the application, how it is being important in the present scenario, especially in the taxonomy and in horticulture and in agriculture, embryology having a lot of importance. It shows very correlation, very prominent role in the classification of the plants, even in the natural system of classification, Bentham-Manuker system of classification, even in the artificial classification, Linnaeus classification. Also, Linnaeus taken consideration of anther structure and number. Anther structure and number. Monandry, diandry, triandry, like that, decandry. How many number of anthers are present? Based upon the anther, he classified the flowering plants. That is sexual classification. And even Bentham-Manuker classification, natural system of classification also, embryological characters taken consideration in the classification. You see, ovary structure, either it is inferior ovary or superior ovary, number of petals, number of sepals, whether sepals are fused or sepals are open, or petals are fused or petals are open, or epipetalous petals fused with the anther, or lacules, number of chambers in the ovule, ovary, number of ovules in the ovary, and types of ovary, and position of the ovary, superior ovary kept in the past and inferior ovary kept in the last. Whereas in the gamma petale, inferior ovary past and superior ovary last. Like that, some majorly flowering characters taken consideration. This all the flowering character will be comes under our embryology, embryological characters. Okay. You can see the anther, what type of the anther it is, monothecous or dithecous, but having the single chamber or two chamber, four lacules or three lacules. How the structure of the anther, type of the tapetum. Based upon the type of the tapetum also classified. Okay, amoeboid tapetum and periplasmoid tapetum, and based upon the pollen structure, microsporogenesis, MMC, microspore mother cell, and anther wall, anther structure, filamentous structure, attachment with others, development of the pollen grains, and structure of the ovule, and types of the ovule, atropos, anatropos, orthotropos, compylotropos, amphiotropos, cercinotropos, ovules. What type of the ovules are present? In the primitive, you know the anatropos ovules in all the gymnosperms. Funicle, chalaja, micropylarian, and embryosac are in the same straight line. Whereas in the developed one, it is carved into 180 degrees and 90 degrees, and modified. The micropyle and funicle comes nearer, and even embryo sac also curved embryos are present in Amphitropous ovules. These all taken consideration in the taxonomy. all the unique characteristics, embryological characteristic plants are grouped into one family, one order, one class. And based upon the sporogenesis, how the tissue sporogenesis is taking place, megaspore and microspore, and ovule, how the megaspore is generating in the ovule, monosporic or bisporic or tetrasporic type, and megasporogenesis and development of the embryo sac, how the embryo sac is development taking place, meiosis and mitosis, mitotic divisions, types of the embryo sac, and mature embryo sac structure, how many number of the antipodal cells, egg cell operators, polar nucleus, that we are discussing yesterday in the polyembryonic. And what type of the fertilization, how the fertilization is taking place, fertilization process, fertilization mechanism, and the carrier, the pollination mechanism, what type of the pollination, how the pollens are reaching towards the stigma, what type of the fertilization is taking place in the plants, how the fertilization process is taking place, any incompatibility, sexual incompatibility is there or not, and endosperm type structure, what type of the endosperm it is, cellular endosperm or nuclear endosperm or elobial endosperm or ruminate endosperm or endospermic aileron layer and mosaic endosperms, what type of the endosperms which are having, that also taken consideration in the taxonomy. And embryo, what type of the embryo it is, whether it is dicotyledonous embryo or monocotyledonous embryo, or it having the cotyledons or not, it having the fluvular radicle or not, or it having filled with the endospermic or non-endospermic, and what type of the seed coat it is, and the testa tegmen is there or not, only testa is present or only tegma is present, or ategmic, if there is no ategma seed coat, or extra seed coats are there as like eril and coreopsis, like that, comos, like that, any extra seed coats are there, that all will be taken consideration in the taxonomy. These all the points is taking consideration in the taxonomy, especially in the Cyperaceae family. There is a variation in the development of the pollen grains that taken consideration. With that consideration, they could able to find out, they could able to separate the Jansianaceae and Cyperaceae. Of course, the microspore based upon the function, and for microspores in the Jansianaceae, this indicating that this Cyperaceae family probably derived from Jansiaceae family. Like that, Shaw done an experiment on Cyperaceae family based upon their microsporic and nuclear status, either it is in four nuclear or two nuclear or three nuclear stage, that are taken consideration. You see in the family Centrospermales order, Cactaceae, having the all the Centrospermales characters. You see glandular antheratapetum shall become 2 or 4 nucleate, 2 nuclear or 4 nucleate with the glandular tapetum. Amoeboid tapetum and glandular tapetum, two types are there, na periplasmoid tapetum. And microspore mother cell, it, two meiotic divisions are succeeded by simultaneous quadripartition of the microspores. Two meiotic division. Usually one meiotic division will take place, and the remaining all mitotic divisions will take place. You see, trinucleate pollen grains. It having the trinucleate pollen grains. Three nucleus are there in the pollens. There is a characteristic feature of the centrosperm. And you see compylotropous ovules, which are strongly covered funicle and massive nucleus, compylotropous ovules, having the compylotropous ovule. These all the characteristics are similar to the centrosperm. Here you can see the ovule is curved, but embryo sac is in state in the compylotropous. That is common in compylotropous ovules, are present common in the pulses usually. You see archisporial cells, it having micropyle form the swollen tip, and the nuclear cap organizing from the periclinal ends, and functioning of the chelagel megaspore, and formation of monosporic 8 nuclei embryo sac, and functioning of the perispore as a main storage region. You know, the perispore means the remaining endosperm after development, after maturation of the embryo. And then disappearance of most of the endosperm mature seeds. Then single layer endosperm will be remain, that we calling it as endosperm. That all the characteristic feature are taken consideration, very minute, even minimal changes that leads to form the different family in the different order. Okay, and in the Loranthaceae also, as for Johri associate, as for Johri studies, Loranthaceae shows embryologically different from Viscoidaceae. Embryologically different. On what way it is differ? Development of embryo sac, endosperm, and embryo in the location of vigil zone. That leads to the what happened? The subfamilies that leads to the subfamilies of these Loranthus. You know the Loranthus that with epiphyte, it grown on branches of that trees, name especially in the name of branches, epiphyte, partial epiphytic, no, the parasite, partial parasite, complete parasite. You might have studied. Trapa, no, that means, we have studied, the Castita, complete parasite. Like that, Loranthus also, that is not the complete parasite, is a partial parasite. Okay. And then you can see the monosporic tetranucleate embryo sac is characteristics of all the Vanagraceae, and but not found in any other family of angiosperms. Okay. And then, but the genus Trapa, it having the 8 nucleate embryo sac, but which was once placed in the Onagraceae, but it's removed. Why? Because it having the not the tetranucleate embryo sac type. That's why the removed Trapa and the Trapa is given to the separate level and separate order. So Trapa does not belong to the Onagraceae family. Based upon the embryo sac character, based upon the nucleus which in the embryo, either 8 nucleus or 16 nucleus, like that, they are separated. You can see variation in the Trapezian Vanagraceae. You see, ovary is semi-inferior. Here is Vanagraceae in the totally inferior. Here it is bilocular. Here is the tetralocular. And megasporogenesis, here chelagel megaspore functions, and it takes further divisions. And usually micropylar megaspore is function and it takes the divisions. And endosperm is locking, no endosperm in the trapezy, but nuclear endosperm is present in the onagraceae. And solenod type of the embryos are there, and vanagra type of the embryos are present in the onagraceae. And you can see the well-developed haustorial suspensions are present, and very short haustoria are present in the onagraceae. And the cotyledons extremely unequal in the trapezy, and two cotyledons are equal in the cotyledons are equal in the onagraceae. And large one-seeded drupe roots are present in the trapezy, and laculicidal capsules are present in the onagraceae. These much variations is there in the family levels also embryologically. Though a constant characteristics are taken consideration for the classification taxonomy, some of the in the phylogenetic classification, not only the morphological character, they're taking consideration of embryology, palynology, anatomy, physiology, cell biology, genetics, molecular biology, genes, sequencing. These all taken consideration in the phylogenetic taxonomy classification. Like that, here embryology is playing key role in the separation of the identification and separation and giving the distinguishing positions to the families up to even up to the genus level. These are more prominent. You see, then palynology. No, we will go for the next. And then here you can see how the relationship is there in relation to the taxonomy. I already told you, according Maheshwari and both, presence and type of anther and tapetum, number and arrangement of anther, and mature pollen grains types, whether it is quadripartition of microscope or microspore mother cell, or two lobes or four lobes, presence and type of the anther tapetum, what type of the tapetum it is, and arrangement of the anther laculi and endothecium. These all the structures, megasporogenesis, origin of spores, tissue in the ovule, fertilization, embryo type, embryogeny, endosperm formation, seed coat, cotyledons. These all taken consideration in the taxonomy. These all playing key role in the taxonomy. Okay, and you can see the few examples of role of embryology in the taxonomy, whether it is dicot or monocot. We can consider it only based upon their cotyledons, embryological characters, whether it having the two cotyledons or one cotyledon. The primary classification of the angiosperm based upon the major embryological character, whether it is dicot or monocot. Embryologically, but having a two cotyledons or only one cotyledon. Anatomically also, even morphologically also, whether it having a top root system or it is adventitious root system. Okay, whether it is isobilateral leaves or and what type of the leaf? Dorsiventral leaves. Okay, like that. And then here you can see so many embryological characters are taking consideration. You can see the Caryopillis, where it is. The Caryopillis. See, this all having the centrospermae. Main embryological characters taken consideration. And you can see Helobiae, monocotyledons order, treated as a subclass based upon their helobial type of the endosperm. You know, types of the endosperm you have studied. Orchideales also, like that, the simple basic embryological characters are taken consideration, like Caryopillidae, You see ambitropous types of the ovules and caryo and no endosperm or little endosperm it is present in the caryopillates. The caryopillates. Okay. And then you can see the helobial type of the endosperm and orchideals, they having the distinguishing embryological character. What it is? Presence of undifferentiated embryo, little or no endosperm, non-endospermic, minute seeds are present. That's why microspermae. Okay, podostomacea. Podostomacea, you see, you're having the pseudo embryo sac. Pseudo embryo sac is present. The distinguishing nucellar structures. So that's why they separated into the families level. And Onagraceae having the onagra type of the embryo sac. And Cyperaceae, you know, from each microspore mother cell, 4 microspores are functional. 4 microspores. Usually only one will be functional, but in the Cyperaceae, the 4 are being functional. In the Laminaceae, you can see helobiales are from the Oraceae, the phylogenetic study. And you can see Crassulaceae type, Crassinucellate. You know, the other rosales are very much nearer to the Saxifragales. And Parnesia, you can see these also, they having the dissimilarities with the Saxifragaceae so that they're separated. Like that, there are so many variations, so many distinguishing characters, very specific characters are there for the identification, for the classification of these embryology. This embryology is taking part in the classification of the plants in the taxonomy, especially the taxonomy. The embryology is playing key role. Embryology is playing key role. And you can see the experimental embryology. The naturally, the pollination, fertilization will take place. That is common. But here in the embryological experiments also done so many lot of experiments has done. That experiment, especially artificial synthesis of the seeds, artificial synthesis of the plants by using the artificial technology like tissue culture technology. Here, the culture technology we are using for the culturing, for the developing, for the production of the new varieties of the plant, homozygous type of the plant, high yielding variety plant, high resistant variety plant, or drought resistance plant, and which are having the dormancy to prevent the dormancy in the seeds, to germinate more number of the seeds, more number of the ovaries. We are following the different culture, ovary culture, ovule culture, anther culture, pollen culture, root culture, endosperm culture, various cultures we are doing artificially. You see the ovary culture, we are taking out the ovary and making it sterile and using in the tissue culture that with that by providing the optimum media, we are producing the female plant characteristics, new female characteristics, plant, fully homozygous plant, we can produce with this ovary culture. with this ovary culture, we also can produce applied plant. Applied plants we also can produce not only by the ovary culture, ovule culture, also by anther culture, anther, pollen, anther or pollen culture by separating the pollen or anther, that we providing the optimum medium to grow that the anther or ovary or ovule will generate new plant. You know the all the plants having the totipotency. Any cell can produce all the plant. That capability is present in the plant. That is called as totipotency. Due to that, from any organism, any organ, we can produce the new organism. Anther, pollen, leaf, root, stem, whatever it is the part, either somatic or embryological. Somatic tissues, that means either root, stem, leaf, or from the embryological, that means the anther, oval, ovary, nucellus, endosperm. From any tissue, we can produce the new plant by this experimental embryology. Gynogenesis, that means female gametophyte. From the female gametophyte, if we produce the new plant by providing the optimal medium to grow, that is gynogenesis. Usually female gametophytic plant, either ovule or ovary. Either ovule or ovary, if we use, that we calling it as gynogenesis. Either through embryogenesis or regeneration from the callus. The both the ways you can produce this plant. Especially we want to grow the haploid. That's why we have to take this ovule or ovary or egg cell before the pollination, non-pollinated. If it is pollinated, it will be diploid. We have to open up the flower and we have to take clean it, rinse it, and we have to remove the all calyx and corolla, and we have to incubate the culture. We have to take it out. We have to make it sterile, and we have to keep it on aseptic media, and then we have to maintain the culture tubes in the dark rooms. You can see the you have to select the flower. That flower we have to wash with the distilled water and dip in the 5% of T-Fall solution and washing with the distilled water and the laminar flow, airflow chamber with, for the aseptic conditions. Surface sterilization with the 5% of sodium hypochlorite solution and then washing out and scalpels. With the scalpels, we can take it out, separation of the ovary, and that separation ovary, we are incubating in the optimal media, which are useful for the growing of the ovary. You can see that this ovary will be germinate and it forming the new haploid plant. There is gynogenesis, either from ovule or from ovary, whatever it is for the female gametophyte, it is from the female gametophyte. That's why we calling it as gynogenesis. We calling it as gynogenesis. Okay. Whether it is pollinated or unpollinated flower, that we should take care. And you know, and simple nutrient medium is 2,4-D or 2,4,5-T and sucrose. 2,4-D and 2,4,5-T, which we use for the culturing of these ovules. And you know, in the tissue culture, what we use, white media and MS media and N6 mediums. Usually we use the MS media. Okay. And then you can see the mechanism I already seen. We have to keep it in the dark room for the 2 to 3 weeks. And after callus formation, you can see, you can separate, and we can uh, make it do it aseptic condition, and then we can go to the uh, greenhouse. We can grow in, and then we can shift it into the outside. You see so many plants which are growing by ovule or ovariculture. Ovular ovariculture, especially gerbera plant that are growing for the in the large level that we are preparing in the ovule or ovariculture. And you know the Hadium vulgare barley and Varieties sativa rice and Triticum vulgare wheat, allium cepa, onion, and Nicotiana tobacco, brassica oleracea, avalu rapeseeds. These all are plant varieties of plants generated through the ovule or ovary culture. Okay, and then you can see so some of the limitations also is there. Number of plantlets per ovary is quite one or two. It will be applied plant. Limited species only it is doing. Only less than 2 dozen species so far. This ovule culture, and then you can see the ovule separation of the ovule from the ovary. Earlier we have studied ovule culture. Now we are studying ovule. Ovule culture, ovule separation of the ovules from the ovary. The separation of the ovule from the ovary. That ovule which having the embryo sac, integuments, nucellas, endosperm. Is all that ovule also we can, especially Maheshwari ovule culture first done by White 1932 in the Antirrhinum majors. Later Maheshwari and Lal also done on so many plants in India, and so many ovule culture plants are generated. Okay, and then you can see the by the ovule culture, how the callus develop, how the new plants are germinated. The ovule culture, especially in the early zygote, 2 or 4 cell pro-embryo stage only we have to take. It is a very important in the boon for plant breeders obtaining seedling from crosses. But in this way, it is very useful. You also can pollinate the test tube pollination, the test tube, whichever the pollens we needed, that only high yielding variety, high resistant varieties. Okay, hybridization in between the two varieties, two viable variety, so that produce the hybrid production of the applied collars by supplementing with IAA oxygens. Ovule culture of orchid plant, orchid very rare plant, they up to the seeds level. Seed capsules of many orchids take long time to mature. It having the long dormancy. It takes years together for the germination. So if we do the ovule culture, we can grow successfully with this separation of the ovule and culturing and growing organogenesis in this ovule culture. With that, we can grow very ornamental orchid plant, especially uh, Podubnaya and Arnoldi, Jan and Calanthe and Cypripedium and Dendrobium plants. We need not to wait still the maturation of the embryo and development of the seed. We can directly do the embryogenesis with this technology. And also it, virus irradiation through ovule culture. We also can produce virus resistant varieties and eye resistant varieties, eye yielding varieties, You see endosperm culture. We also can separate endosperm from that endosperm also either cellular or nucellular endosperm or relobial endosperm. Uh, Endosperm culture that is very useful in the formation of triploid plants. Triploid, usually the endosperm is triploid so that the endosperm is using for the triploid and sterile seedless varieties. That is done by Lampe and Mills at the earliest, and Nakano successfully cultured immature endosperm in the rice. Rangaswamy and Rao also done endosperm culture in the Shantalam. You see, immature endosperm and mature endosperm, both you can separate the endosperm. You see that the maize is easy yellow color part, especially in the grains, wheat, maize, paddy, especially in the cereals. It's easy. And you can see incubation of the culture, embryogenesis. What are the factors? Nutrient medium which was needed, low nitrogen, and which will be useful for the production of triploid plant. It shows the self sterility and increase the edibility. So there's no seeds. Those are so there are seedless fruits like grape, banana, watermelon, and also timber and fuel yielding plants. It shows better performance. It shows the better I percent of the timber. And then pollen culture, isolation of the pollen from the anther and make it sterilization. The applied embryonic tissue, the pollen. You see, Simakura has done the first the pollen culture. You see, Tukali and Ginkgo and Guha and Maheshwari also done the pollen culture in the Datura Inaxia. You can see how the pollen culture, the steps are there. You have to take the anther, you have to take the bird. That bird we have to remove the sepals and petals, and we have to take it out the anther and discard the filament. The anther you have to gently crush the anther in the glass jar and filter the pollens with the mesh and pollen suspension. We have to say make it separate and then resuspension again on MS board and pellets of the pollen that we have to take it out and incubate in the petri plates under the MS media at 24 to 26 degrees centigrade up to 3 to 4 weeks so that that pollen will multiply and it form the embryos. That embryo is forming from the pollen. That's why it is only applied. It having all the plant characteristics. There is no extra other plant characteristics. Why it is homozygous from the same plant we have taken. And by for the production of haploids, there is a very good method, pollen culture, pollen or anther culture and ovule culture is a good method for the haploids productions. And endosperm culture is good for parthenogenesis. Okay, and then you also can see the various culture, float culture and nurse culture methods and androgenesis. What are the nutrient medium MS and niche medium? Niche first done, that's right. And usually you see we have to take the consideration of the time, season. What are the factors affecting? Activated charcoal, temperature, stage of anther, photo period at what time you are taken, the flowering time, endogenous agents, and age of the plant. These all characters affect the pollen culture. So many importance are there for the cytogenetic studies, genetic recombination studies, uh, differentiation in the single cell organism, and how the factor, what are the factors that controlling pollen embryogenesis and mutagenetic studies, and in the plant breeding and the applied tissues, applied preparation, especially Arabidopsis, Lycoperdon, are used to transfer the E. coli gene transfer. You can see the root culture from the root also, somatic culture, Katte and Robin White. White first successfully cultured tomato roots. The White, in the 1934, first culture successfully the tomato from the tomato roots. From the root, they are generated the plants. You can see the root tips we have to take fresh and dry way, the medium, and these all are effect. And you can see how this culture is taking place, how this genesis is forming. You can see here how the root, the separation of the root, sterilization and filtration and growing it under the culture inoculum, and then it grows, it forms the new plant from the root. So many important, especially in the root crops, especially that in the for the root crop, growing of the high varieties, high yielding varieties, and high quality varieties of the uh, root crops production. These are very useful. And shoot culture also, from the shoot, apical shoot also, we are growing. You see glycine marks and Phaseolus vulgaris. These are all grown in the root culture, having high yielding varieties, and regeneration of shoot and root. You see synthesis of secondary metabolites, airy root culture. With that, we can produce the root based production, roots based. You see, there is a different type of the roots product, especially datura, like burstum, paisum, in this all, with having the various type. Yeah, these all are the applications and experimental embryology, various cultures, root culture, stem culture, embryo culture, anther culture, ovule culture, ovary culture. This all embryogenesis. Huh? This all by using this, by maintaining the aseptic condition, by providing the optimal medium, we can produce new varieties of the plant. That all experimental embryology is now playing key role in the horticulture and in agriculture. Is playing big role in the horticulture to produce varieties of the ornamental plants and to produce various crop yielding plants, fruit crops, root crops, and various type of the flowering plants for the production and the maintenance of the homozygosity, maintenance of the mother genetic material. These are uh, stopping of the heterozygosity and maintaining the genetic purity. These all experimental embryology is very taking part much. Okay, and then the next is the palynology. Till now, we have studied embryology, importance of embryology in the this one uh, in the taxonomy. Embryology important, and then we will go for the palynology. What does it mean? The palynology. Palynology means study about the pollens. Study about the pollens. Palynology. The palynology word first. coined, paleontology word first coined by, what is that? Hyde and Williams. Paleontology word first coined by Hyde and Williams. Hyde and Williams first given the name term paleontology to study about the pollens. What it is? The study of the pollen or spores. You know the word paleontology, it came from the Greek word palen, pollen means sprinkle, spread, sprinkle one. Whatever it is, spread, sprinkle, come out, small sprinkle, it come out from the plant flower. What it is? The pollens will come out, sprinkle, come out from the flower. That's why they are given the name of pollen. Okay. You know the study of the pollen, structure, importance, principles, prospects, these all comes under paleontology. Okay. You can see the pollen or spore, the various type of the pollens, having the various aperture structure, very ornamentic structure, they're having the odd coated, non-stylatic, non-hydrated. The toughest coat is the sporopollenin exine, outer layer of the pollen. Due to that, it could able to survive for the long time and even resist to the abnormal environmental conditions, uneven environmental conditions. They are the characteristic feature of the pollen. Pollen is a male gametophyte, male gametophyte, the male gametophyte. Microspore, also called as microspore. You see, the first pollen microscopically observed by Nehemiah Grew. Okay. The importance of the pollen in the spermatophyte plant, first done by Robert Brown. And Geopart first described the fossil pollens, fossilized pollen. And Cunningham and first report the illustrated account of the airborne bioparticles. In the air, what are the pollens are there? The pollen grains, fungal spores, insect fragments, whatever it is that all observed by Cunningham and Sharp first. Microprotograph of the spores pollens has done. You know who is the father of paleontology? Who is the father of paleontology? I already told at the starting. Gunnar Ertmann is the father of paleontology. And who is the father of Indian paleontology? I already told you P.K. Naik. Who is that? The paleontology has an interdisciplinary study. It having the much more importance even in the taxonomy, embryology, horticulture, agriculture, in the so many studies. These all paleontological study, pollen study also taking consideration in the so many other branches. You see, not only it is restricted to the pollen grains and spore study, it is interdisciplinary study. It deals with the study of extant and extinct polymorphogenesis. So many types, so many variations that all consideration in the taxonomy. Applications of paleontology in taxonomy. Okay, that is paleotaxonomy. How the pollen structure, what type of the pollens, what is the variation, how it is spreading, what is the ornamental structure, how it evaluated. These all taken consideration in the taxonomy. So paleontology is showing much importance along with the embryology in the taxonomy. Okay, and then you can see aerobiology or allergic study. In the aero applications, lot of applications are there for palynology. Palenotaxonomy. By observing the pollen, with the pollen also, we can classify the plant by the palynological study. And aerobiology, aeropalynology. What are the pollens are there in the air? What type of the pollens? How many type of the pollens? Whether it's allergic pollens or normal pollens? Uh, that all. What are the pollens which are there in the air? That aeropalynology. So many applications are there for the palynology and melitopalynology pollens which are there in the honey. Melitopalynology honey. We can find out, we can categorize the honey, whether it is pure honey or not, by observing the pollens which are there in the honey. If the pollens are present in the honey, that we feel it is a pure honey. If the pollens are not there, we can't say it is pure honey. And also by studying the pollens, what which plant pollens are there, based upon that we can see, we can categorize, we can know the purity of the honey. From which plant flower the bees extracted the honey. Whether the more number of the pollens of sunflower or hibiscus, whatever it is there. So we can feel it is collected from that plants. And one major aspect of the palynology is forensic palynology. In the criminal investigation, in the forensic science, in the criminal investigation, in the forensic science also, pollen study is playing key role. The observing the pollens on the dead body where they are died, and also observing the pollens on suspected people. Suspected people, if he feel he is the killer, he cause for the death of that person, we can observe the pollens upon suspected people and pollens upon the dead body or died people are victim on the victim and also suspected people, so that the same pollen, if it is there, you can easily identify as like DNA fingerprinting. We also can find out the exact criminals by observing the pollens. So many histories are there, pollen study criminal cases. By observing the pollens, so many cases are finalized. Like that, the pollens also playing key role in the forensic study in the criminal investigations, where the criminal kill the victim in the forest area or in the flowering plants area or in the open area or in the mesophytic area, mesophyte plants area or xerophytic plants area, where what type of the pollens are there in that area, that all taken consideration to identify exact criminal, exact person. And also biostratigraphy, that means bio geochronology, that we the by studying the pollens, we can see, we can know the year, how long it is. You know, the pollen may not die and dry. It will be remain even for the million years. If even in the all the plant may die, all the plant may be decomposed, may not be fossilized, but pollen will be definitely will be fossilized. By studying that pollen, we can know how long these type of the plants are already existed, how long back these are originated, how from since how long these all are getting, these all are surviving. That all we can know by this palynological study. Why? Because the sporopollen in hard-coated tissue is present, that an enzyme is made up of its sporopollen. Due to that, it will not degrade. And even in the paleopalynology, fossilized plant studies also, I already told you, the past vegetation, what type of the plants are there, that also earlier environmental conditions, earlier climatic conditions, also in the we can know by studying the pollens. Okay. Also improve in the crops improvement also it will be very useful. You can see much of applications is there in the geology, the geological studies, the paleo environmental studies, earlier environmental climatic condition to know the earlier environmental climatic condition by observing the pollen in that area, and geoengineering, geoarchaeology. By observing the pollen, we can know what type of the fossils, what type of the plants might be there at that era. And even archaeological studies, fossil plant archaeological studies also we can know. And human died oldest era, in that era, what type of the human died maybe. And paleobotany, what are the fossilized plants are there? You know the Birbal Sahani Institute of Paleobotany, the Lucknow, study about the fossilized plants that especially pollens is very much easy to find in the fossil than the plant other plant pot. Even in the oil industry also we can use this pollen. That means what type of the plants they compressed and it cause for the formation of the coal and crude oil. You know the what we are getting out coal and crude oil, that is because of olden days plants that are fell down and compressed, fell on the earth and it compressed, it is deep compressed, it formed the crude oil, and is it the more compression, it formed the coal. That all are because of that we are all getting that crude oil and coal, that area means olden days, that area is fully covered with the dense forests. The dense forests are collapsed, that all compressed and that all fossilized, that form coal ball certification, and so much of the fossilization has taken place. That fossilized plants only from that fossilized plant material only we are getting the crude oil and coal. You see, Earthman is the father of paleontology, and father of our Indian paleontology is P.K.K. Nair. Who is that? Parameshwara Krishnan Kurti Nair. P.K. Nair is our Indian paleontologist. Paleontology depending upon four characteristics of the pollen. What are those? Whether it is having the resistance to degradation, why it having the I already told it having the I resistance then to the degradation, then the remaining parts of the plant is having the show I resistance. Why it having the sporopollenin? The small in size, very minute, and transport and deposit sedimentary particles is very easy, and morphological complexity is much so that we can easily distinguish, and the plant they can produce enormous number of the pollen the flowers, so that easy to identify and characteristics. You know the pollen character, pollen apertures, pollen structure, various type of the apertures. You can see porate, colpate, colporate, porous type, non-porous type. You know, you see NPC system of classification based upon the porous characters, number of pores, position of the pore, and characteristics of the pore. You see the pollen it having the outer layer, pollen wall is sporoderm. The sporoderm having the exine and intine. Exine is mainly consists of the sporopollenin, which are electrolysis and decay resistant. But remain for the long time. Okay, so that very easy to study paleontological study and forensic study, archaeological study, These are sporopollenin. Because of the sporopollenin, it is very easy. You see the pollen structure, sporoderm structure, exine and intine. Exine again divided into two, that is sexine and nexine. Sexine is ectosexine and endosexine. You can see, and nexine again divided into nexine 1, 2, 3, ectonexine and endonexine. You see how the pollen wall structure, how it is, how they modify. You can see in the sexine again, it having the tectum and bacille. The porous cavity space is called as bacille. The rod shape structures are called as tectum. Tectum and bacille is there in ectexine, and foot layers is in endexine. You can see intine, ectointine, and endointine. Intine is made up with pectocellulose. Exine is made up with sporopollenin. You see tectate, columellate, folate wall. You see the tectate type, how the apertures are present. You can see the ectoexine, it having the tectum and columella bacilli. The sculpture ornaments, you can see the intine, how it is, the colpus, whether it is having the sulcus or colpus or porate. What type it is, narrow porous or lesion, narrow or porates, how it is. You can see trilate, 3 layer, and monolate, only one, or the various type of the pollens, monocolpate, tricolpate, various types are there. These all are, you see, the pollen characteristics, mainly 7 categories: aperture type, pollen wall, and pollen unit polarity, symmetry, size, and shape. The pollen morphology is taken consideration first for the identification, categorization, distinguishing. You see the symmetry, whether it is showing the symmetry or not, what type of the aperture it is, how the exine and intine, tectum, baculum, intexine, endexine, how it is present, and the pollen arrangement, whether it is mono, di, tri, tetra. In the tetra, whether it is decussate or T-shape or linear or polyar shape, or a nemac pollens, whatever it is. You see, either it is polonium, or like Orchidaceae and Asclepiadaceae, whether it is showing the polarity or not. These all these all taken consideration in the pollen study. So many principles are there: polarity and shape, whether it is distal polar end is large or uh, partial polar is larger, so that we can easily categorize. And isopolar or heteropolar, whether it is showing the symmetry or asymmetry, what type of the shape, side, oval, rectangular, ellipsoidal, uh, what type of the aperture it is. These all taken consideration. And you know the Ettman classification, Ettman, father of palynology. He classified the pollen based upon the NPC system of classification, that you know, number of fours and position of the four and characteristics of the four. Okay. Trim, aperture. What type of the aperture? How many number of the aperture? Where it is the position. Trim, you see monotrim, ditrim, tritrim, tetratrim. You can see, but it's only single pore, mono, di, tri, tetra, penta, hexa, and poly. If it is more, if it anamo, more are arranged. Irregularly arranged, that is anomotrim. And in the position, built upon the position, whether it is distal side or proximal side. Usually proximal side kata, if the both the side ana kata, only distal side ana, jono if it is the middle, if in the zone 2 rows, the dijono, if irregularly arranged, that is penta. And the tree, if it is having the colpate or porate, you see if it is a forest type, that is lepton. If it is having the two tree, that is tricolpate. If it is having the arrow type, that is colpate. If it is having the both colporate. These are the characteristics taken consideration in the classification of the pollens. Okay, and and we also can see the various type of the pollens. You can see the how attractive the structure, stem structure, acacia pollen, hibiscus pollen. Uh, you see how attractive tricolpate it is there in the cypress. You see monoporate in the graminae. These are winged pollens. Polyporate pollens that are there in the hibiscus. You can how hibiscus pollens you can see how attractive, and dentella pollens. These all are there. How the aperture types and size are there. These are all type of the apertures, ornament, external. You see how attractive it is. Okay, and then where we are. There is not. Okay, and then here you can see the palynology in relation to taxonomy. We already studied pollen aperture, size, shape, structure. This all taken consideration in the taxonomy. Different, we can distinguish, differentiate between the two various type of the pollen storage products. How it is, that all we can see very easily. What type it is, how it is, the pollen study, the pollen units, how in the pteridophyte and gymnosperms, how it is, what type of the variation. You can see the pollens in angiosperms and gymnosperms. There is a much variations. These all are the applications of palynology in the taxonomy. Okay, these all are various types, various studies of the pollen and palynology, importance of the palynology in taxonomy, and applications of palynology in various branches. Okay, melatopalynology, forensic palynology, geopalynology, paleotaxonomy. Hmm? These all are various types of the palynological applications are there. Much of the importance of their pollen types, pollen usage, based upon the pollen characters. These all are taken consideration in the taxonomy. With this, we are concluding the. With this, we are concluding this. We almost all completed all the syllabus. Medicinal plants 2 units and embryology 3 units. We all cover almost all the syllabus, 99% of the syllabus. You all go through these YouTube. You watch again and again. If you have any doubt, you can ask the questions. You repeatedly you see question wise. If suppose importance of palynology in taxonomy, that much only you listen first once, and you can get the outcome. What is the importance of the palynology, and what is the applications of the palynology? What are the palynological applications in the various fields, embryo, in the taxonomy, in the various branches, how the palynology is important. That all also you can see applied palynology, paleopalynology, geopalynology, palynotaxonomy, aeropalynology, melatopalynology, and then lactopalynology. That has caused so many diseases, suspected domain, and pharmacopalynology, the pollen spores which are present in the drugs and tablets, forensic palynology in the criminal branch cases, and copropalynology which are there in the dung, cow dung and waste, excretory material, what are the pollens are there, What are the biogenic pollen? What are the biostatic? That means biochronology. Also, we can know these all applications are there. These all are the applications of the paleontology. Applications of the paleontology, various branches of the paleontology is applications of paleontology, using the paleontological knowledge in different branches in different studies. That is application of paleontology. Okay, there is a question, application of paleontology, and another essay question, embryo application, importance of embryology in taxonomy, importance of embryology in horticulture, and then the experimental embryology, different cultures, ovule culture, different organ culture, ovule culture, ovary culture, anther culture, pollen culture, root culture, leaf culture, shoot culture. These all the culture, but these all are experimental embryology. These all taken consideration in the experimental embryology. That also important. Not only the natural embryological process, artificially also we are generating the new plants by using